Morning. Seems like we have much fewer people. Perhaps, perhaps everybody's in the long weekend mode. I don't know, including tomorrow's holiday. Anyways, we'll get started. So today we're going to look at influenza viruses. So what about influenza viruses? Okay. So this is something that we all should know. A lot of times we say, I had flu, she had flu, or somebody in the hostel had flu. But usually that's often used as a misnomer or used inappropriately because you may not have had influenza, but the term flu has come into play. Usually it's used loosely and is associated with fever, body ache, anything you have, something like that. Just say that I had flu, but that's not correct actually. But when the true influenza is when you get infected with influenza viruses A, B, or C. I'll show you, I'll tell you more about these. So influenza viruses belong to a group of viruses called orthomyxoviruses. And like I told you, there are three influenza viruses, just like polio virus. 1, 2, 3, there are influenza viruses A, B, and C. And influenza viruses are larger than polio viruses. I'm just comparing it to polio virus because that's the only other group of viruses that we did in great detail. So they are negative sense RNA viruses and they have 8 segmented genomes. Remember, I told you that some viruses, especially only RNA viruses, can have segmented genomes, and influenza virus is one of them. Just like we have chromosomes, multiple chromosomes, they have 8 segments in their genome. Instead of having just one genomic stretch of RNA, they have it broken down into 8 segments. And every time there is an infectious virus, it needs all the 8 segments to be packaged, and that is the infectious virus. Okay. All of you know that it causes respiratory infections. It can also cause other symptoms which are systemic. That means it's blood spreads through blood and can cause it symptoms in other parts of the body like muscle ache and fever and other things which are generally referred to as systemic symptoms. Okay, here we go. This is what influenza virus looks at if you look at it up close. So we'll do it one at a time. Perhaps we can start at the center. Like here are the 8 segments that I told you about. They're not equal length, they are of varying lengths. And each one typically codes for one protein. So usually that's how, usually, I mean, basically it has 8 proteins. Okay, there could be some other smaller variants, but it has, mainly it has 8 proteins from the 8 segments. Okay, you see this little yellow dot at this end of all the segments? What do you think that is? Okay, it's written here. Okay, that's the RNA polymerase. There are 3 subunits of the RNA polymerase called polymerase basic unit 1, 2, and the acidic unit. Now, what do these 3 subunits do? If you're interested, you don't necessarily have to by heart it. So one of them acts as a RNA polymerase. Okay, so that just copies RNA. That's this one. So what do these two do? They have interesting functions. What is the modification of most of our mRNAs at the 5' end? Anybody? Our host mRNAs, human mRNAs. Guys?
Can you repeat the question?
Okay. The question is, what is the modification of most of our mRNAs or most of the RNAs that we have in our body, particularly mRNAs? What is the modification that happens to our or human host cell mRNAs at the 5' end? Go ahead. Go ahead. Sorry, I didn't hear you.
5G capping.
So that's called 5G capping. That's correct. So there is a
Capping,
cap.
capping of guanosine.
That's correct. So there is a cap that's called a guanosine cap that's attached to the 5' end of many of our mRNAs. And what is the function of this? It protects, it has many functions. Very quickly, the 5' caps help increase the half-life of the mRNA by preventing RNAs mediated digestion. That's one thing. The second thing that they do is they also facilitate nuclear export of mRNA. So mRNA, because all see mRNA is messenger RNA, and obviously we want proteins out of it. So mRNA is synthesized in the nucleus, but proteins are made in the cytosol. So it has to export mRNA. All our mRNA are made in the nucleus, and they have to be exported to the cytosol. And the cap also facilitates that, and that's the 5' guanosine cap. So most of our mRNAs have it. Now, these two subunits of influenza virus, what they do is they have the ability. So this one PB2 has the ability to bind to the cap. That's our cap, 5' guanosine cap. This binds, and this can actually cleave it, and the acidic subunit PA can cleave it. It has endonuclease activity. It can cleave the cap from our mRNAs, and then they can stitch it to their mRNAs or their RNAs, so that the stability is increased. So this is called 5' guanosine cap snatching, and a handful of viruses are adept at it. They are very skilled in doing it. They borrow or just snatch the cap from our mRNAs, and that is possible by these three subunits. I'm just going to clear it because it's become too crowded. So these three subunits. So any case, that's just extra detail. You don't necessarily have to by heart exactly what I told you or anything like that. That's just for information about what these RNA polymers. So each unit has its own RNA polymerase complex. Okay. And these are called, because they are protein and RNA complexes, what do you think they are called? Anybody? They occur in human cells also.
Ribozyme.
They're called RNPs or ribonucleoproteins. Okay, so that's what they're called. So the same term is used for these fragments because they protein and RNA is attached together. They're called RNPs or ribonucleoproteins. And then I'd like to draw your attention to this blue layer right here. That's called a matrix protein. Now, we haven't looked at matrix proteins in viruses as yet in this course, but a good number of viruses have matrix proteins. What is the function of the matrix proteins? Now, the function of the matrix protein is primarily to be an interface between the capsid proteins, which are inside here, and the envelope, which is outside. So it's an interface between the capsid and the envelope. In between these two, many viruses could have a bunch of proteins called matrix proteins. Okay, so this is like more like a supportive layer, but it has many more roles other than just supporting. I'll tell you a little bit more about at least one key function of the matrix protein in just a bit. So that's this protein right here. So that acts as an interface between the inside, which is the core or the virus capsid protein, and the outside, which is the envelope. That's the role of the matrix proteins. In general, in most viruses, that's the role, but they have many additional functions. We'll look at it. At least one we'll look at it. Then you see this little channel-like pore here, which is called M2 protein. This M2 protein is nothing but an ion channel. Do we have ion channels in our cells? Anybody? Guys?
Yes, sir.
Yes, sir.
Okay,
Neurons combines and
Yes, sir.
that help in like propagation of the action potential.
that's correct. Nervous impulses. And we have ion channels. We have plenty of ion channels in our cells, and they have a variety of functions. We do have ion channels in our cells. But viruses can also have ion channels. I'll tell you what, this is a peculiar kind of ion channel. I'll tell you what is peculiar about it and what's the function of M2 in just a bit. So that's this matrix protein. It's just like a pore. Sorry, this is the M2 protein. It's like a pore or an ion channel. Okay, I'll tell you the function in a bit. Then you see something called nuclear export proteins. So these are important proteins that allow these RNAs, which are which are usually synthesized in the nucleus in this case, they have to be exported to the cytosol, and this is the virus encoded protein that can actually allow the export of virus nucleic acids from the nucleus to the cytoplasm, and that's also packaged inside the virion. Now, the last two things that are left are this purple colored stuff and these mustard colored stuff. Now, maybe we'll first look at the purple color. First, I want you to notice that the purple has, like for every 3 or 4 purple, there is only 1 mustard. Okay, the purple is nothing but the hemagglutinin, which all of you are aware of, and it's an important protein for influenza virus. And the mustard colored stuff is neuraminidase. I'll tell you what these two are in a bit, but you must have heard about H and N configurations of influenza virus. Can anybody tell me the name of an influenza virus with the H and the N configuration? Anything that we've heard about often in the news?
H1N1.
Perfect. So H1N1. I'll also tell you what this H1 and N1 means. In a bit, I'll tell you what the H and the N configurations mean. I'll tell you what they are in just a minute. So this nucleic capsid is helical. So what does the helical nucleic capsid mean? Anybody? What is the major difference between the helical nucleic capsid and the icosahedral nucleic capsid in terms of the size? Anybody recalls anything? So one of the important things is
will be bigger than like helical nucleocapsid will be bigger than icosahedral.
What you said first is correct, actually, more or less. So what happens is, if you go back to the slides and look at it, the virus structure slides, I have very little there, but I made this a point to list this in my slides, that for helical capsids, the protein or the protein coat of the virus is being synthesized along with the RNA or the DNA. So where the RNA ends, protein coat will also end. So it's more compact. So what you said first was correct. So these are more compact ones because the size of the helical nucleic capsid is usually determined by the size of the RNA or the DNA, which is the genetic material of the virus. That determines the size. Whereas icosahedral, actually, it doesn't have to be really smaller genomes, have to have smaller capsids. There is no rule like that. They could be a little bit this side, that side. But here, because they are closely associated with the nucleic acid, the size of the nucleic acid determines the size of the capsid in helical capsids. Okay, now I'll tell you a little bit more about what we refer to as the H or the hemagglutinin. Now, what is the function of the hemagglutinin? So first thing, it's a trimer. What do we mean by the word trimer? Anybody?
Made up of 3 monomers.
Very good. That's correct. It's made up of 3 monomers, which constitute this protein. 3 similar units constituting 3 units together make this protein. That's called a trimer. And that is exactly why, if you look at this cartoon here, it has 1 bulge, 2 bulge, and the 3rd bulge. So hemagglutinin is a trimer. Okay, so what is the function of the hemagglutinin? So this is the part of the virus that latches on to the host cell, and all of you know that it latches on to the respiratory epithelial cells, including the cells of the lower respiratory tract, because we get lower respiratory tract infection with these viruses. So any virus that we are concerned about in terms of respiratory viruses are usually the ones that cause lower respiratory tract infection, because if you get mild infection of the throat, sinus, all these, the body can take care and it's not life-threatening. But whereas lower respiratory tract infection, which is the infection of the bronchi, bronchioles, and the tissue, lung, etc., is is life-threatening, and it can cause pneumonia and a lot of other things. So anything that we are concerned about, any virus that we are concerned about, in terms of respiratory tract viruses, they always attack the lower respiratory tract. Okay, so that's one part. So this is the virus protein that's responsible for latching on to the respiratory tract epithelial cells, and it latches on to something called sialic acid, which is abundant on many cells, including respiratory tract cells. Now, anybody knows what is sialic acid? So these are carbohydrates. Sialic acids are made up of carbohydrates. They could be linked to glycoproteins, but they're basically carbohydrates in nature. Okay, so now it's not just the virus latching on to the sialic acid residues on the host cell surface is just not adequate to let the virus in. There is one more very critical step that happens, and that happens by host cellular proteases. What do we mean by this? Anybody knows in terms of SARS-CoV-2, everybody's heard about something called ACE2, right? What is this?
Receptors.
ACE2 is a receptor for which virus? I mean, in terms of viruses, it can do a variety of host functions. Let's not talk about it. I won't even know much about it. But in terms of virus functions, what do you think ACE2 does? Why do we all know ACE2? It's a receptor for which virus?
Coronavirus.
That's correct. But in terms of coronavirus, have you also heard about something like this? SS2. Have you heard about something like this transmembrane protease? The function of something like this in coronaviruses. Anybody heard about anything, read about anything like this? No? Okay. So I'll just tell you what it is. Go ahead, go ahead.
Yes, so sir, I read about that it helps in the priming mechanism of the spike protein, which further
That's
facilitates
correct.
the viral entry.
That's very correct.
In addition to other proteases also, so cathepsin and all.
That's correct. So there are a bunch of proteases that are present in our respiratory epithelial tract. And these proteases, what they do is that they have the ability to cleave certain residues in a specific motif. So this one, the one that I just wrote down, it's called transmembrane protease. That's why it's actually helping the virus entry, because it's just is part of it is outside, part of it is inside. That's why it's called transmembrane. That's the TM part. It's a serine protease. It recognizes a specific moiety or a sequence and then cleaves it. That's the function of this. Now, why is this important? This is important because without the cleavage by this protease, hemagglutinin is not able to complete the function of virus attachment and penetration. So what do I mean by that? So just sticking to the sialic acid receptors will not let the virus in. It will not. Same is true for SARS-CoV-2. Just sticking to the ACE2 receptor will not let the virus in. Like one of you rightly pointed out, there is this mechanism called priming of these virus proteins. The term priming refers to nothing but actually cleavage by the host protease. And after this cleavage, they become separate subunits. And one of the subunits is able to actually actively allow the virus into the cell, because that after this cleavage only fusion can happen, something called fusion with the cell membrane, and entry can happen only after these host proteases cleave it. So accidentally, it turns out that accidentally we have the right receptor, we have the right proteases, both of which are important to allow the virus into the respiratory tract. This is true for influenza. This is true for SARS-CoV-2 as well. Now, so there could be different subtypes of hemagglutinin. There are different sequences of hemagglutinin. I'll tell you more about it in the next class, which will be on Friday. Tomorrow is going to be a holiday. So I'll tell
Sir,
you what.
please explain the previous point again.
Yeah. Which point? The cleavage part.
Yes, sir. Yes, sir.
Okay. So maybe very quickly I'll try to tell you what the cleavage part is. So maybe okay. So here's hemagglutinin. It's I didn't want to tell you all this because you asked. I'm just going to go into a little more detail. So the typical hemagglutinin that comes with the virus, which is uncleaved, that's called HA0 or HA0. And when these proteases cleave it, I'm just going to put it protease or serine protease. Okay, when they cleave it, it becomes actually something called HA1 and HA2. Now, this HA2 has the ability to actually fuse with the membrane, host membrane, and then let the virus in. So there are some conformational changes that happen because of this cleavage. And without those changes, the virus will not get into the cell. So this cleavage by this host protease is critical for the virus to enter the cell. What is the host protease cleaving? It's cleaving the hemagglutinin into separate units, not necessarily 3 units. It just does it. Don't worry about that part. It cleaves it into HA1 and HA2, and the HA2 has the ability to actually, this conformational change that it brings about, this cleavage, allows HA2 to actually complete the fusion and get into the cell. Without this cleavage, that won't happen. The same thing analogous to this, the spike protein has S1 part and it has a S2 part. Okay, this is a spike protein of SARS-CoV-2. And pretty much one of these proteases or a bunch of proteases are important to cleave the S1 from S2, and ES2 has the ability to actually fuse with the receptor better. And this conformational change allows it to fuse, and then is when it can get in. This is what triggers the penetration of the virus into the cell. And without penetrating the cell membrane, there would be no infection. Is that better? Do you understand a little bit better?
Yes, sir.
Okay. Okay, I was telling you about the types of hemagglutinin. I'm not going to go into the details today, but I'll tell it to you in the next class. So there are a total of 18 different sequences of hemagglutinin, which are considerably different from each other. Like you can see, they're called subtypes. It's more like the virus subtype itself. Okay, so remember, viruses had genotypes and genotypes had subtypes. And there were serotypes and there was quasi-species. That's what we looked at in one of the lectures, I think the last one perhaps. So this is actually, if you look at major different types of hemagglutinin sequences that are available till date, there are 8, you can classify them into 18 different HA types based on the sequence. Of course, in HA1, not all the viruses have identical sequence. There are going to be quasi-species in this. That is not what we are talking about. If you classify them like the phylogenetic tree concept, they will club into 18 clusters, and they're called 18 subtypes of hemagglutinin. Okay, and in humans, we have only a few subtypes that infect humans. There are a few more additions to this list. Perhaps we can try and talk about a little bit more about it in the next class. So these hemagglutinins, the name should tell you that if any of you are wondering why they're called hemagglutinins, they were named a long time ago, and they were named because like several decades ago, scientists identified that this purple protein on the virus surface has the ability to bind and clump RBCs of specific species. What do I mean by that? Let's say I take influenza viruses of some kind, or okay, I take a virus, maybe influenza virus of a certain kind, and I take a particular species of RBCs, okay, red blood cells from, let's say, monkeys or chicken or some animal like that, and I just add it. all the RBCs will clump together, and that was brought about by this protein called hemagglutinin, and that was the name. That was the reason for the naming of this protein as hemagglutinin, because it has the ability to agglutinate heme containing cells or RBCs. So, or make them stick together. Now, anybody wants to guess how this happens? Very simple guess just from the slide. Anybody? RBCs are rich in sialic acid too. They have the receptor that this can bind to. So they have the ability to make them clump together because they just have, it's a lock and key mechanism, right? Receptor and a ligand. So hemagglutinin can bind to sialic acid and RBC surface, and because see, because there are so many hemagglutinins, they can clump different RBCs, at least a few RBCs together, and one virus can do a few and another virus can do a few. They can just form something called a lattice or cross-link RBCs, and that agglutinase RBCs, and that is the reason for naming this protein as hemagglutinin. Now going on to the second component, which is the N configuration that we spoke about, the H1N1. This is the N in the N1. So neuraminidase is the name of this protein. I'll tell you what it does. So it's a tetramer again. So that's why this cartoon has 1, 2, 3, 4 subunits. It's a tetramer. Obviously, you might have also noticed that it's less frequent than the hemagglutinin on the envelope, maybe 3:1 or 4:1 in that ratio. More abundant protein is the hemagglutinin. Neuraminidase is less abundant. Okay, now it's a sialidase enzyme. That's the chemical property of this protein, and it can cleave sialic acid residues. Now, why does the virus need it? Okay, so here is a part of the answer. There is another part of the answer. There are two parts to the answer. Why does the virus need neuraminidase? These are the two reasons. One I'll explain to you today, another one perhaps in the next class. Now, this part I'm going to try and explain very quickly to you guys today. So most of the like at least the respiratory epithelial cells, they can secrete a mucus film. Mucus film contains several things. It's a gel-like substance, all of you know that, and that lines our respiratory tract, and it has important functions in immunity. What does this mucus film do? It's a thin film of mucus that will be there, that the moist part of your respiratory tract. This is all, we have this mucus secretion. This mucus, give me a second. This mucus can trap microbes, and I'll show you a cartoon wherein it can actually push it out. Okay, so that is the function of this mucus. This mucus is also rich in sialic acid. Okay, so if the influenza virus gets trapped in the mucus, there is no way it can do it can reach the lower respiratory tract and infect, or it may not even get a chance to infect human cells. It may not survive. So this is the virus's way of negotiating the mucus trap. It has an enzyme that can make the mucus less viscous, or basically cleave the sialic acid and make the mucus non-mucus or less viscous. Okay, that is the one key function of this protein here called neuraminidase. Now, the second function, this part I'll try and explain to you with a cartoon. I think in the next class we'll do this. You'll understand why better when I show you the cartoon rather than just looking at it like this. In terms of how many types of neuraminidase are there, genetically there are about 11 different types, like 18 for hemagglutinin, and 11 for neuraminidase. Only some of them are in humans, not all of them are in humans. Okay, so this is just a cartoon, nothing much. It's just to show that how it binds to the sugar side chain This is the sialic acid residue on the cell surface. It binds. Okay, now going on to this slide, I'll tell you three things only in this slide, I think. What is the function of the M1 protein, M2 protein, and the NS1 protein? Okay, that's what I'm going to try and tell you in this cartoon. So first, the M1 protein. All of you know, I want to remind you what the M1 protein or the matrix protein is. If I just erase everything, this is the matrix protein that forms an interface between the capsid and the envelope which is in between them. Now, what is the function of this matrix protein? It's very important for virus assembly. So why do we say it's very important for virus assembly? Again, I'll go back to this cartoon. So it has two sides, right? If you think of it as a protein like this, it has one side. Let's just call this outer side. And there is an inner side to this protein. Okay. So this protein, the inner side, has high affinity for these ribonuclear proteins. It has high affinity for that. The outer side has high affinity for the virus glycoproteins, which are hemagglutinin and neuraminidase. That is the outer side. So this is important because this is what allows the virus to see everything gets packaged. This is the matrix protein. These are the H and the N that are waiting on the cell surface. And because this matrix protein has high affinity to the hemagglutinin and neuraminidase, they are picked up from the cell surface and become a part of the virus. They stick to it. And they're also important for the assembly of the segmented RNA because they have high affinity for ribonuclear proteins. So that's why this is very important in the virus assembly. It allows on one side RNA ribonuclear protein packaging. On the other side, it has high affinity for the hemagglutinin and neuraminidase, which are already synthesized, which are encoded by the virus. The virus has the genetic material. The proteins, hemagglutinin and neuraminidase, are made using the host cell machinery, right? Our protein synthesis apparatus it uses. And they go and sit and wait on the cell surface for the capsid to come along with the matrix protein. And because the matrix protein has high affinity for these, when this buds off, it buds off with hemagglutinin and neuraminidase. So that is the role of the matrix protein. Okay, now what is the role of the M2 protein? I want you to focus here, the M2 protein. So once the virus is entering the cell, after attachment to sialic acid, after hemagglutinin binding to sialic acid, and the cleavage by the host proteases, serine proteases and other proteases, it's able to enter the cell. And inside the cell, it's in a compartment called the endosome. This path is followed by almost most viruses. And all of you are aware that inside the endosome, there is a process called acidification where the pH becomes lower. Now, what is the function of this M2 protein? This M2 protein is here. It is an ion channel protein. Again, I'll show it to you very quickly here. This is the M2 protein. Okay, this is the M2 protein. It's a channel through the virus. What is the function? I'm going to tell you right here very quickly. So it selectively allows the transport of H plus ions or hydrogen ions to allow more acidification of the virus. It selectively allows it in this direction from the outside to the inside. So that what step is this going to aid in? Anybody? I already gave you the answer. Anybody wants to say anything? What is it going to facilitate in the broad steps of virus replication? Guys, are you able to hear me or?
Could you repeat which steps?
Okay. So this matrix protein, this M2 protein, it's an ion channel protein that selectively allows hydrogen ions to flow from the outside to the inside. What do you think the function is going to be? This happens inside the endosome.
To maintain the pH.
Maybe the
pH will resolve the coat and also that RNA can come out and replicate.
That's correct. It allows actually disintegration or disassembly of the virus so that the RNA can go out. So that is what this facilitates by acidification of the inside of the virus inside the endosome. It allows more and more H plus ions, makes it more acid, allows it to disintegrate. That initiates, that's a critical step to allow virus replication. That's the role of the M2 protein. Now there is a non-structural protein called NS1. This NS1 reduces interferon response. I'll talk about this more in the next few minutes. We have another 20 minutes at best, 15 to 20 minutes. I think that should be enough. Okay, so how was influenza virus transmitted? So all of you know that aerosol-based transmission, whenever an infected person coughs or sneezes, he brings out a lot of viruses in droplets, and that could infect others. In addition to droplets, surfaces act as a possible medium, like if I'm infected, if I cough like this, and if I switch on the switch and you come and switch on the switch, and then you put your hand into your mouth, from me to the switch and from the switch to you and then to your mouth, it can be transmitted through surfaces as well, because the virus is able to survive a few hours outside. When we say survive, it just retains infectivity. It doesn't have life, or it has life. It's a contestable thing. Anybody can argue about it, but it just retains infectivity for a few hours outside the body surface. And incubation period is like 1 to 3 days. Okay, so what is the function of the neuraminidase? I already told you that it helps the virus negotiate the mucin layer, because the mucin layer contains something called sialic acid, and this is a sialidase, an enzyme that can cleave this and make the mucus less viscous, and prevents the virus from getting trapped in the mucus. That's what the function of this protein is. There is also an additional function which I told you I'll talk about in the next class. Right? Okay. Now, this is all there. I think this we spoke about. This also we spoke about. So, what are why does influenza virus cause damage to the lower respiratory tract or the cells of the lungs? Why does it cause damage? Because one is the virus can actually may cause directly. It can, by replicating inside a cell, it can cause damage to the cell. There's something called interferon-mediated cell death or interferon-induced apoptosis. We won't talk about too much about it, but we'll talk more about interferon, because this virus has the ability to induce a lot of interferon, and that is good for us because interferon appears to be the most important player to fight off influenza viruses. Okay, for each virus that may be different. For this virus, one of the important reasons why we get rid of the virus is interferons. I'll tell you a little bit more about it. And there is also cytotoxic T cells. Can anybody tell me one line about the role of cytotoxic T cells, which are also referred to as CD8 T cells? One line about it, if you know. Guys?
They kill the virus infected cells.
That's all I wanted, actually. So they have the ability to identify and kill virus infected cells. That is the role of cytotoxic T cells or CD8 positive T cells. That's what they do. So that is another reason why influenza infected cells can die or lung cell damage can occur. Now, if you in this cartoon, what I have is if you were to look at the inside of our trachea, okay, if you just cut it open like this and open it like this and see what's inside, this is how it will appear. You have, what are these hair-like structures? Anybody? Guys?
Cilia.
They're called cilia. I'll tell you what they are, and many of you know what they are. These are hair-like structures, and this is uninfected trachea. Within 3 days of influenza virus infection, you can see that the top layer is almost gone, and it slowly starts coming back after 7 days. And to go back to this state, uninfected stage, it will take about 3 to 4 weeks, almost 28 days it'll take to go back to this kind of trachea. So that's what it does to our trachea. There is cellular damage. Now, influenza virus is associated with reduced mucociliary clearance. What do we mean by this? So this is the, if you look at the inside of your trachea, these are the hair-like structures, and this is the mucus, the gel-like substance that contains sialic acid, and that has the ability to trap microbes. And these cilia are always beating like this. If you were to think about your trachea when you're sitting down like this, they are always beating towards the mouth in all of us. Why? Because if anything is stuck, they don't want it to get into the lung. They want it to go to the throat. Either you swallow it into your stomach or you cough it out. So that is the idea of mucociliary clearance. They trap microbes that are trying to enter your lower respiratory tract, and they push it out by beating in one direction. And this is reduced because the viscosity of this mucus is reduced. The ability of our trachea, we are not even talking about, when we say this, we are just saying that the ability of the mucus and the cilia to trap and push out microbes is reduced. That is what we mean. Why is that important? Because a lot of times when you have, in 3 days, it becomes like this. In 7 days also, it's not grown back. The cilia are still, very few cilia are there. So it's important to understand that when you have reduced mucociliary clearance, we have a risk of what we call as secondary bacterial infections. Now, what does this mean? This means actually that your primary infection here is the influenza virus. That made your trachea, the inside of your respiratory tract, look like this. And you've lost the cilia, you have less mucus. And because you don't have much cilia and you don't have much mucus, your respiratory tract becomes susceptible to what is referred to as other bacterial infections, which are secondary to your influenza virus infection. Primary cause is this. This is secondary to that. So they're called secondary bacterial infections. Now, what do we mean by this? Very quickly, we have a variety of pathogenic bacteria in small quantities in our throat. It's possible, like pneumococcus, which can cause pneumonia, is also present in many of our throats, and we are healthy. But when this happens, when they slide down into the lower respiratory tract, there is no cilia, there is no mucus to trap it and push it back. Now you can get infected with some organisms that are present in low numbers in our throat without causing disease. Now they acquire the ability to cause disease because you have reduced mucociliary clearance. Now, how long do people shed when they get infected? 1 day before they become diseased or show any symptoms, and up to about 5 to 6 days after having symptoms, almost about a week of shedding. Okay, this is kind of similar in SARS-CoV-2. It may vary by a couple of days, but pretty much for most respiratory viruses, that's important. Now, I told you, I emphasized that this is a key player in recovery from influenza viruses, which is called interferon response. That's what we look at in the next 10 minutes, and we'll close after that. So specific antibodies to influenza viruses appear in the blood in about 10 days to 2 weeks after you get infected. This part you've already seen, the superficial layers are destroyed, and it takes almost a month for complete repair, this is how the cycle goes. This is where we start, 3 days, 7 days, and almost 1 month to go back here. That's what it means. Okay, so today we'll just close after we look at what is interferon response and how it protects the other cells. So in the next 10 minutes, that's what we'll do. So let's assume this is one of your respiratory epithelial tract cells. It gets infected with the virus. What it sends out once it gets infected, it secretes all these pluses, or whatever this red colored stuff. This is all interferon. It secretes interferons to tell the neighboring cells that a virus is around, you guys just watch out and be ready to fight it. That's what it tells the neighboring cells by secreting this interferon. Now, how does it tell it by doing that? Eventually, it confers what we call as antiviral states in the neighboring cells. Although this cell may die, it may protect the neighboring cells. Now, how does it confer the antiviral state? It does not prevent the virus getting into the neighboring cells. It cannot prevent, but it makes these cells alert and makes them look out or watch out for the virus so that they are ready to fight it as soon as it comes, and that prevents the cell death in these cells, the neighboring cells. So that is what interferon does. How does it do it? So this is the interferon secreted from the infected cell. Many cells just give me 1 second. Hello? Vijika, or 10 minutes la pantuma or lecture mudichittu pantuma. Okay. Sorry about that. Okay. It's all about a lot of relatives coming down with COVID. So anyways, so this is the interferon that's secreted by this infected cell. And the neighboring cells have receptors for interferon. This is the neighboring cell surface. It's much bigger. This is a receptor on the cell surface. It binds to the interferon secreted by the infected cell. This is what comes out of the infected cell. This is an uninfected cell that is getting warned that the virus is around just by this interferon. Now, how does it happen? Now, this interferon binds to this receptor. And once this happens, there is a cascade of events that happen downstream to this. And that induces making of some particular host proteins in large quantities. Okay, so what happens when this cell tells the neighboring cells the virus is around? It secretes interferon, and that interferon is bound to receptors on these cells. All of these cells have receptors to interferon. They bind to it, and that leads to a cascade of events which trigger the synthesis of some proteins in these 4 cells, okay, uninfected cells. Some proteins are synthesized and kept ready. Okay, they're made in larger quantities than usual. Now, what happens after that is when the virus actually enters, I told you this will not prevent virus entry. The virus will come, it will infect. Influenza virus will come and infect it. Remember Baltimore classification? What is the, do all RNA viruses have a double-stranded RNA step? Yes or no? Yes?
No, sir.
Okay, I wish I had the slides. They do, because see, if it's a positive sense, it can directly code for mRNA, but it does make an intermediate negative sense strand. Same for negative sense, it makes a positive sense strand. So both 2 strands of RNA are present in all RNA virus infection. So there will be a double-stranded RNA. You go back and look at the slides. If you have questions, you can ask me back again. Okay, it's very simple. It's very evident from the Baltimore classification slides. Otherwise, also you can imagine it. But if you have questions, you ask me in the next class, no problem. So this will happen when influenza virus infects this cell. This will happen. When actually influenza virus infects the cell, it turns on some proteins. The body has the ability to sense double-stranded RNA because we normally do not have any double-stranded RNA. We have negligible amounts of double-stranded RNA. So when there is a large quantity of double-stranded RNA inside the cells, the body has a mechanism to sense it. And once it senses it, it can activate some host proteins, which can either degrade the RNA, or it just shuts down the entire protein synthesis, including our own protein synthesis in the cell, so that the virus proteins cannot synthesize. And once the virus RNA is gone, then it turns back on our protein synthesis so the cell can survive. So I have a little bit more detail here. I think this will be the last slide for today. Or do I have more slides? I think that's all. I think this will be the last slide. Okay, now When the interferon from the neighboring cell binds to the interferon receptor in the uninfected cell, which are the proteins that are made and kept in large quantities? Remember this cartoon? Some proteins are made in large quantities before the virus comes, and the cell is ready to fight the virus. Which are these proteins? Usually these three proteins, maybe a bunch more that we don't know. One is called 2' 5' OAS, or oligoadenylate synthase. The other one is a ribonuclease. The other one is a protein kinase. So I'll tell you what happens. So these proteins are synthesized and kept in large quantities in the uninfected cells. Once the virus infection happens, you have double-stranded DNA that this cell can sense. Pretty much all our cells can sense the existence of double-stranded RNA. I said DNA by mistake, it's double-stranded RNA. So this comes from the virus. Once the virus actually infects the cell, this happens. And then this gets activated. And this activated 2' 5' oligoadenylate synthase can activate another protein called ribonuclease L. Obviously, ribonuclease has the ability to degrade all RNA inside the cell. This protein is activated. Now, virus RNA will also be degraded. Our mRNA will also be degraded. So this double-stranded RNA, which comes from the virus, also activates protein kinase R, which can add a phosphate moiety to a protein called eukaryotic initiation factor 2, which is important to start any kind of translation or protein synthesis in the human body. This is very important. Okay, this anchors the tRNA to the ribosome, and it's important for that. If this does not happen, protein synthesis will be shut down. No more proteins will be made in the cell. The cell simply refuses to make protein, and by shutting down protein synthesis of the cell, there is some damage to the host cell, but no virus proteins can be made, and virus RNA gets degraded. Once the RNA is degraded, it won't be able to replicate. Then this protein synthesis, everything is reversed, and the cell can survive. Okay, so this is how forewarning with interferon secretion by infected cells can save the neighboring cells. Okay, although they will get infected. Now, we have just 2 minutes. So I told you I'll tell you one more line about this. So this is a virus encoded protein called non-structural protein 1, influenza virus encoded protein. It has the ability to bind double-stranded RNA, which is primarily going to be influenza virus RNA. When it's replicating, it will be double-stranded. Okay, for those of you who are not sure about whether it's double-stranded or not, you can go back and look at the Baltimore part. But believe me, it's double-stranded. Now, once it's double-stranded, this is pretty much the only double-stranded RNA inside the cell, and this virus protein can go and coat it or bind to it. And this binding prevents our sensors. We have something called toll-like receptors, or TLRs. I think I've shown you one cartoon, maybe, or maybe not, it doesn't matter. But these are receptors inside our cells and outside our cells that can sense things that are foreign, pathogen associated molecular patterns. You don't have to by heart any of this. Whatever I'm saying now, it's not written in the slide. You don't necessarily have to do it. They just identify some patterns. They identify unique signatures of pathogens, including double-stranded RNA, which is usually not there in the cell, can be sensed by a toll-like receptor. Now, this toll-like receptor does not recognize double-stranded RNA when NS1 is bound to it. So thereby inhibiting interferon response. If interferon response is inhibited, the virus survives better. So it's already encoding a protein that will help it survive better and beat the interferon response in the host. This protein is encoded by this virus, which encodes only a handful of proteins, right? So that is the role of the NS1 protein. I think that's where we'll stop. Any questions in today's class? We have maybe maximum 2 more minutes. Guys, any questions? Go ahead.
Sir, doesn't NS1 interfere with viral processes involving dsRNA?
Say that again. Which one?
Sir, like the non-structural protein one, when it binds to double-stranded RNA,
Yeah,
so it cannot be recognized by host TLRs, right?
that's correct.
But doesn't it interfere with the viral processes? Like it had to process its mRNA and all. from the dsRNA step.
Say that again. Viral processes.
Huh.
Like what?
So like virus has to encode its own proteins now from the RNA.
That's correct.
So when this NS1 is bound to the RNA, wouldn't it interfere that process as well?
I don't think so, but I can find out and tell you. I get what you're asking. You're asking whether it will inhibit virus replication.
Yes, sir.
As best as I know, not, but I can try to find out more. I mean, there is so much of literature. I can try to find out more. But what I know is that it just binds to double-stranded RNA and minimizes interferon response because TLRs don't recognize it as good. But I'll look at what you asked and I can tell it to you in the next class whether it also interferes with interferon virus replication. Usually there are not many proteins like that, but anyway, I can look at it and tell you. Anybody else has any other questions? Guys, seems like today everybody is very quiet.
Not
Either...
right now, sir.
Okay. Okay, then we'll close for today. Thank you, guys.
Thank you, sir.
Thank you,
Bye.
sir.
Thank you, sir.
Thank you, sir.
Bye.
Thank you, sir.